Well, hello everyone. I'm Sofia, and I'm here today to present the work that I've been doing recently in the team that I'm working with. To begin, let's talk a little bit about what is Rett syndrome. Well, Rett syndrome is a rare genetic disorder that is characterized by a mutation in the gene MECP2 that codifies for a protein with the same acronym. This is a very interesting syndrome because 95% of the patients diagnosed with this condition are female and is characterized by four distinct stages. For our team, the main focus is the late deterioration stage because this is what leads to the early death of most of the patients. Oh, well, that was an accident with my little thingy pressing the button, but let me move the mic a little bit and here. But let's talk a little bit about the complexities about Rett syndrome because here we have a lot of things to talk with. To begin, remember the 95% of the patients diagnosed with this condition being female? Well, for a long, long time in this condition, most of the research was made in male mice model. This is an issue particularly for Rett syndrome because MECP2 is codified in the X chromosome, which is distinct for the stochastic silencing of genes that occur in the regulation when two copies of the X chromosome exist. This led to the patients to having a very heavy mosaicisms in all of their bodies with a direct correlation between the distribution of MECP2 positive cells and MECP2 negative cells having a direct impact in the phenotypic severity of the patients. And this being an stochastic process indicates that there is not a precise marker that we could use to see how, this will develop later. Another big issue with Rett syndrome study is that MECP2 is a transcriptional regulator that works in both repression, activation, chromatin loop formation, and chromatic compactation, which means that when it doesn't function properly, most of the effects that happen for its dysfunction had a very wide cascade of effects on the body that we have not a very good understanding on how they work. And given that most of the research was done in male model, we also do not understand how the patients being female affect the condition really. To tackle this issue, we decided to do an analysis on single cell. For this, we use actually a public data set that is from the study identification of neural oscillations and epitheliform change in human brains organoid. That consists of an organoid dataset made from Rett syndrome patients that is composed of a combination of human cortex and ganglionic eminence. From this, we did the traditional preprocessing with Seurat, and we performed separately a trajectory analysis of the samples to see how the development process may change between the MECP2 positive and negative cells. From the most interesting groups for the focus of our study, we did a regulatory network analysis from which the most significant results for the condition were modeled into Boolean models to further explore the dynamics that may happen in this condition that may be useful for future treatment. The first result that we did was the trajectory analysis. This trajectory analysis is said to be with a pseudo time based specifically on developmental stages of the samples, which means that these groups are not separated by if they are MECP2 positive or negative, just by the developmental time points within the samples. We have 31 groups, and what we could see on this trajectory analysis was that despite this being a pseudo-time based developmentally based trajectory analysis, there was still a distinct separation of the MECP2 negative cells, with them taking some sort of what we describe as shortcuts, because in some of the developmental process, particularly on the number 4 and 19, they need to reach from 0.7 to 0.25. This step takes 3 steps for the normal cells. However, the MSCP2 negative cells, of course, the MSCP2 negative cells tend to take 2 steps, which is something that was previously recorded for this condition, that they tend to develop hastily, but there also have not been further studies that would show that this process does happen. It was an hypothesis, but here we can see that they tend to take a few developmental steps to try to reach the same point, but they are functionally distinct to the point that even in cluster with the same pseudo-time developmental time point, they tend to separate within the cluster themselves, like if there were oil and water in a single glass. The 2 main groups, we start the regulatory analysis with dopaminergic neurons, since we are more interested on the later stages of the syndrome. And given that this is a syndrome that develops science peer, we try to focus on more developed neurons for this process. We started with dopaminergic neurons, where we could find this system of master regulators, which are active in both conditions, with marked in yellow. And what's interesting about the dopaminergic neural network is that we have some targets that are non-coding RNAs that appear in the sample that are different for MCP2 positive cells and MCP2 negative cells. This was really significant for the condition because there has not been like a direct situation where non-coding RNA can have been like connected to this, but this was very puzzling, given that most of these non-coding RNAs are not described, for which we could not further infer what the biological process was of this, but the fact that there was a distinct positive and negative target non-coding RNAs was particularly interesting for this condition. However, our next group of interest, the GABAergic neurons, gave us a little bit of more straightforward answer. Here we have a system of master regulators for GABAergic neurons that are possibly co-regulating each other. We have a system that is active mainly in MCP2 positive cells in orange, and a system, a postulated compensatory mechanisms that activate in MCP2 negative systems. This was particularly significant for the condition because we have some markers that are phenotypically relevant, and we have 2 genes that indicate that this system could possibly be a non-cell autonomous system, which means that this system is not activated when there is a mutation in MSCP2, but rather by the cell-to-cell communication and regulation processes that may happen in this type of cells. From this, we decided to further explore the dynamics within the samples, specifically the GABAergic neuron samples. For this, we did the dynamic Boolean network modeling of the samples using the binarized time series of the GABAergic neurons to further explore the distinct states that they may have, and possible insights in the phenotype and the dynamics that may reach to that phenotype. And we also did this to further explore the difference between the two. Given that I was not very sure that the video will work, this is a static summary of the most significant changes that we can see. We have 3 main static attractors. We have 3 main static attractors that are highlighted in blue, that are active in both conditions and are very significant for both of them. We have MCP2 positive attractors that are distinct and significant for the MCP2 positive cells, which include MCP2 and DLX5, which phosphorylize MCP2. And we have some distinct attractors that are active in the absence of MCP2, which indicate that our proposition that this model could be a system of main regulators and compensatory mechanisms. Those holds up, as well as key targets that have phenotypic relevance for the condition. As general conclusion of what we have done is that the set-of-type calculation enables us to see how the developmental pathways differ between the MCP2 positive and negative cells. that we postulate that there may be a system of non-coding RNAs that has an impact on the functionality of the dopaminergic neurons. However, this needs to be a lot more explored biologically because there has not been much of in-lab research about this topic. We also proposed the system of master regulators for GABAergic neurons that is exclusive to MCP2 and, well, healthy neurons, as well as the compensatory mechanism that may be active in the MCP2 negative neurons that are co-regulator each other at checking each other at different steps of the cycle, as well as the dynamic properties of the system, both validates the proposition of the main and compensatory mechanisms, and of course, and that this dynamic could be key for further understanding how Rett syndrome may work, and especially for late-stage Rett syndrome patients. And that will be my presentation. So I'm open to questions if anyone has any.
Thank you, Sofia. We have time for one question from the public. No? Well, I have one. Sofia, I have a question related to the non-coding RNAs you found. Can you say which type of non-codings, long non-codings, or any kind of?
Yeah, we checked which type of non-coding RNAs they were. They are not currently very described, but most of them are long non-coding RNAs, which was particularly interesting because MECP2 has a regulator, does have a tendency to particularly police, if we can say it like that, long non-coding RNAs. So this may explain the difference in targets that we may see, but we need to validate with a biological model to see what is exactly going on with non-coding RNAs.
Okay, thank you, Sofia.
Thanks.